So we've talked about the origin of life and the origin of viruses. Now let's talk about a smaller subset of this, the origin of coronaviruses. Because why not? Yeah, just because it's interesting. And it'll give you some extra grist for the mill for one of the test questions. So, if you look at coronaviruses, and if you find new coronaviruses or corona-like viruses, you see some interesting things. So, shown here are the maps of the polyproteins of some coronavirus relatives. And so what you can see here is that they're all set up in roughly the same way. So instead of having one big polyprotein, they have a polyprotein that's broken down into two chunks. A open reading frame 1A, which seems to always contain this blue thing, which is actually a relative of the main protease, and an open reading frame 1B, which always contains the polymerase and the helicase, so the equivalent of NSP12 and 13 of coronavirus. And if you look beyond that, there is pretty much nothing else that these things have in common with coronaviruses, but they are the closest non-coronavirus group to coronaviruses. And a couple of these are published, all the ones in black. You can go and look up their genomes and play around with them. Yeah. And so the idea is that these are to be called the Protonida That's the most likely sort of finishing point for their name. And so they're going to be related to the Nidovirales, which is the group that contains coronaviruses. All right, so looking at these, you can see that they have other things in common with each other that they don't necessarily have in common with coronaviruses proper. Now, let's look a little deeper. So we can look also to viruses that are a bit closer. So these would be within the Coronaviridae, within the coronavirus family, but in various, mostly insects, I believe. And the bottom one's a spider. And so once again, we've broken down the genome. The coloring is the same, and so you still have the blue thing here, which is a main protease, but now it's flanked either side by these little black bars, which are transmembrane regions. Places where you see a sort of a broken edge like this, that just means it's a partial genome, and we don't know exactly what comes before that. And so that's the open reading frame 1A. Then we have an open reading frame 1B, which is going to contain the NSP12, RNA-dependent RNA polymerase, and the stripy thing now, that little candy cane there, is now the NERAN domain. And so everything inside the Coronaviridae actually has both a NERAN, well, it has a NERAN hooked up to its NSP12, which means that they probably all share a common way of starting virus genome synthesis. Now, another thing that you find is this helicase down here, which is the dark purple solid region, which has a conserved neck, which is just a connector, and a conserved, these little sort of ovals that are white. Those are zinc fingers. So this is a metal binding domain, and you find it in every single coronavirus to some extent. Here's a weird one that only has one in there, and it's kind of missing the neck. And then after that, you'll find some of the proofreading enzymes. So you'll find the yellow is our proofreader, the blue is one of our methyltransferases, and the gray at the end is our second methyltransferase. So the equivalent of NSP14, XO, and N7 methyltransferases, and the gray is the NSP16 equivalent. So everything in coronaviruses, or at least the majority, seem to have these parts attached to them. And zooming out to the broader corona-like viruses. So actual coronavirus, the only representative is going to be this one at the top, SARS coronavirus. And you can see some of these other viruses which are related. And they all have the NERAN, our little stripy pink bit. And they all have the NSP12 polymerase, RNA-dependent RNA polymerase. They all have the zinc fingers, which is just a metal binding, zinc metal binding domain, attached to the helicase, which is the purple. And mostly, but not all, but mostly they seem to have the two methyltransferases and the exonuclease for error correction. The one group that does not have these are actually the smallest of the corona-like viruses. They're called arterioviruses. And yeah, weirdly, they're missing both the capping system and the proofreading system. So that's pretty crazy, right? But yeah, nevertheless, they persist. They exist in the world. You can also look on these. We have the rest of the genes up here, and everywhere that there's something like a spike, I've colored it sort of a salmon color. Everywhere that there's a nucleoprotein like N, I've colored it green. And some of these viruses have things that are like M that I colored orange, or some of these viruses have a thing which is related to a different group of viruses, alpha viruses. Chikungunya would be an example of an alpha virus. Some of these can cause really bad hemorrhagic fever, but they have a different sort of thing. Instead of a spike, they have two copies usually of the same gene that will interact with each other and will do the same job as the spike, but they're called a class 2 membrane fusion protein. And they work quite differently from the class 1s. They start out flat as pairs. They stand up and rearrange so that there's actually 3 of them that come together, about like that, in a not quite pyramid. It looks about like my fingers are showing. And they have a fusion peptide at the tip. And then once they have that, they'll actually lay back down, interact with a different part, and so that is effectively doing the same thing, bringing the membranes of the virus and the cell together, and then they can enter. Now, in some of these viruses, we can see that there are proteins that we can't find. So here is NSP3. It's from the yellow all the way up to the brown. NSP4 is this white thing with 4 transmembranes, and NSP6 is this little, it looks black because it has a lot of very close together transmembranes. That comes right after. These are the 3 that make the DMV pore, and make the DMVs, actually. And you can see that those exist in coronaviruses. They might exist in toroviruses. It looks like we've got reasonable relatives. Possibly exist in arterioviruses, although it's getting pretty shaky. We can only find a few parts. And for the rest of the corona-like viruses, we cannot find very good matches for these. There are things that could be them, but they're not close enough that you can be sure. But take one of these. This is the abyssovirus, which is, there we go, in Abyssoviridae. And so it has this thing. And so it's basically uncolored because I only colored in the parts where we can match them up. And we actually looked inside the cells, and lo and behold, what do you see? See those? Those are little virions. Those are virus particles that are just forming. And see these around them with a little stringy stuff inside, and looks like 2 membranes going around the edge? They're a little weird, but those look like double membrane vesicles. And they're in a cell that's definitely infected. Look at all that new virus coming out. You can even see a lot of these viruses in the process of being formed. They're sort of still flat, and like here's one that's working its way to becoming a virus. So this is a distant coronavirus relative, and it also appears to make double membrane vesicles. And if you have a double membrane vesicle, there have to be pores. And so, yeah, it's just a matter of locating the pores. But probably these proteins, which do not appear to be related, are actually going to end up doing the same job. It's going to be a case where there's too distant to find nucleotide or amino acid homology, but you can still find some structural and functional homology. Another example of this, if you compare arterioviruses to coronaviruses, just 2 members of the group, you will find that coronaviruses have one M protein, and it's going to be involved in the membrane. You can actually see the M is making these little connections here between the membrane going down. And I don't know if you can see it, but there's usually 2 little blobs, kind of like a figure 8, that sit on top of each of the connections. If you refine the image from a whole bunch, like thousands of them, it actually looks about like this. And let's actually move that over a smidge. There we go. So in coronaviruses, the M proteins, you get 2 copies of the same M protein that come together and make that little, well, I guess it looks more like that. It's pointy at the bottom, structure. And that's the thing that joins the nucleoprotein inside to the spike-like proteins on the outside. Arterioviruses don't have spikes. They have a set of 4 proteins that together, we don't know what they look like, but they come together and all 4 look different, and they're all involved in this, and somehow those 4 do the job of a spike. And it is still a complete mystery as to how they do this. So they don't really have a spike, but they have something that works like one. And they do have a nucleoprotein that's actually related to the coronavirus nucleoprotein. But if you look at their membrane, you can see, boop, boop, look at those, the little dots stacked up. There we go. And so basically, it's just a slightly smaller, shorter version of exactly what you find in a coronavirus. So even though these are distantly related viruses, once again, and even though you can't spot the similarity between these proteins, they're actually doing the same job. And the neatest thing is that whereas the coronavirus is 2 copies of the same protein, the M protein, in arteriovirus, this is actually made of 2 different proteins. There's one called M and one called GP5, and they interact, and they actually are disulfide bonded together. And darn it, but they make the exact same structure that's doing the same job. So they are probably homologous to the coronavirus M protein based on structural similarity. And in looking at coronaviruses, we can actually also identify several other points of similarity. So, non-structural protein 8, remember the golf club? Here's the head, here's the shaft up here. We can find this in every coronavirus, so it's definitely there. At least all the proper coronaviruses, but in the other members of the nidovirales, the corona-like viruses, we have the shaft part, but we don't have the head part. And the head part is actually where the enzyme is, that's the primase. So it looks as though in coronavirus relatives, they either have a different primase as the sort of middle member of the chain, of the team, or they have some other way. Maybe their NERAN is enough to get started and polymerase can just take over from that, main polymerase. Who knows? Someday we'll know, yeah. So this long alpha helical region appears to be conserved. And we know this forms the exit tunnel on the back of the polymerase. This is where the RNA comes out. It's like a long tunnel that protects it. And also, the other thing that you'll find conserved in pretty much all of these is a thing that I would call the metal binding landmark, the nimble. This is going to be a little protein that's equivalent to NSP10 in coronaviruses and binds 2 zinc molecules as mostly beta-stranded and is located right at the end of polyprotein 1A every single time. And it's always located just after the NSP8 equivalent. So these are things that look similar at the level of structure or predicted structure in these cases, but you cannot find any similarity at the RNA level or at the amino acid level. Yeah. The last little parts here. So we had a lot of those alpha virus-like spike proteins popping up all over. Those are the ones that were sort of light blue a couple of slides ago. These guys, and they see alpha 1 and 2 on them. Generally come in pairs, but there are a few cases where it looks like one may get used twice. Yeah, because there's only one gene there. These are actually pretty common in different viruses. They make a fold like this. You get them in the Bunya viruses. And Cenorhabditis elegans, the little worm, has a similar fold in it. So it looks as though this may be a fold which was stolen either by life from viruses or by viruses from life. There's our alpha virus version, and flaviviruses like yellow fever virus. They have one as well. As do hantaviruses, which are very bad for you. Yeah, you don't want to get one of those. So, and we would add on some coronavirus relatives may have this. So you can look at the family tree and you can go down it and say, okay, all of you members of the tree, what do you have in common? Because the idea is that if most branches have something in common, then you can spot where a new thing creeps into the genome. And so let's move this over a little. There we go. So to do this, you look at the tree topology. And so here, every branch, so these are some of the branches of the corona-like viruses. And our coronavirus branch are these guys, orthocoronavirinii. Those are the normal ones. Like SARS-CoV-2 would be in this branch. And so here we can see that, okay, viruses like SARS and like its closest relatives, the proper coronaviruses, all have a blue thing. So they have a nucleoprotein with two domains, an N-terminal domain that has a particular form and does a particular job, and a C-terminal domain. Different shape, different job. But you can also see that across the nidoviruses as a whole, that is unusual. All the rest of them have this green thing, which is the C-terminal domain of nucleoprotein only. So from this tree organization, from the tree topology, so we're looking at time, starts over here and then goes to the present, which ends in a little dot or the end of a triangle. We can see that probably if the early branches have a C-terminal domain and the next branches have a C-terminal domain, and the next branch after that has either an N-terminal domain and a C-terminal domain, or just a C-terminal domain, then probably the ancestral condition, the original, last universal common ancestor of all these things, the thing that would be at this node right here, that probably had just a C-terminal domain. Yeah, and it's just logic, and it's hard to go back and actually prove, but you look at enough of these genes and you can start to build up a picture of corona-like virus evolution. And so, here we go. What kind of spike do we see in these groups? And so, all right, we have class 1, which is going to be regular spike like you and I know and love. And so here in the middle on those branches, yep, we have what looks like spikes. Down here, we have what looks like spikes. But the earliest branch point, every single one of those has an alpha virus-like protein. And once we get to this branch point, you end up with this side. Most of the things that go this way have an alpha virus-like protein. Some of the recent ones show what appears to be replacement. It looks as though the old alpha virus-like protein got replaced with a more spike-like protein. Yeah, so that's neat. Over here, this side appear to have spikes, but when you go too far down this branch, you get to the ones that are like arterivirus, that have that split 4-part, who knows what it is, protein. And these just have unique solutions which do the same job as the spike, but are not genetically related to the spike. So you have another replacement, probably a recombination that picked up another gene that just happens to have worked, but viruses probably try this all the time, and it probably almost never works out. So these are very rare events. But yeah, they would be game changers for the virus. Okay, and let's look last little chunk here at the M protein, which is normally embedded in the membranes, that 3-pass transmembrane protein of coronaviruses. And you can see that we have M-like proteins only on this sort of middle branch from arteriviruses over to corona. And you can see that those all actually do form a clade. They're all downstream of this branch point where my cursor is hovering right now. Everything over here does not have an M. All of these super early relatives in the protonidoviruses do not have an M. Therefore, M is not part of the original set. So, weirdly, the coronavirus group, the defining features of the virion of this group are actually pretty different in coronaviruses compared to pretty much all of their relatives. These coronaviruses that we know now are, they're weird, yeah, compared to the group of viruses that they belong to. I think that's quite interesting. And you can actually go further. You can look and say, okay, do we have a main protease in all these viruses? And looks as though we better move this up a little bit. Here we go. Let's put that on front. And let's make this smaller. Let's move it together, and then we'll be good. There we go. Sorry, you have to see me editing like that. There we go. But yeah, main protease, yeah. It is ancestral because it is in every single one of these groups. These are the main groups that branch off. RNA-dependent RNA polymerase that's related to the picornavirus RNA-dependent RNA polymerase most closely. Yep, you bet, every time. Now, weirdly, NSP9 is only found in coronaviruses, which means that all the rest of these, if they use a protein-primed replication and transcription method, they must have some different protein that they attach it to. So there's nothing fundamental about NSP9. It's just the latest thing that's like a little brick that you can attach a nucleotide to, and whatever, it'll bind to a part of the genome, and yeah, it'll work. But that appears to be, rather shockingly, a variable in corona-like virus evolution. Helicases, yes, and always the same type, always there. Near-RAN domain, no. Only the real close coronaviruses. It is a derived trait. And so A means ancestral, means they have like the early version, like if our tree looks like this, then they would have the version of things that came first. Actually, let's shrink our... Oop. Oop, 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 oop. Nope. Let's see if we can shrink. No, we can't. Okay, never mind. Yeah. And so you go through and say, okay, there's always a set of transmembranes that occur right before the main protease, so the transmembrane 2 region. But the transmembranes don't always occur after this, and it appears that in the early branches there are none there. So this is a new trait. The helical landmark that's like the shaft of NSP8 does appear to be there every single time. And there's always apparently an envelope on the virions. So they're the same general type of virions. They're just swapping out their proteins that actually make the virion by the look of it. And the last thing that looks as though it is ancestral, completely ancestral, is the N-terminal domain, or rather the C-terminal domain of the nucleoprotein. There you go. So that's a little walk through corona-like viruses and how you can figure out where these things came from, who's a super early relative versus just a kind of early relative. And I know the questions that regular people outside this class tend to ask usually focus on, well, where exactly did this virus come from? Who was the person? What was the place? Give me an hour and a minute. But we can also take these kind of big picture views. And it's really useful for just getting a better understanding of what is a virus? What's a virus do? What's a virus have to do? What can you change and have it still be a virus or part of a group of viruses and still work, basically? And yeah, the answers are surprising. So there you go. Thanks very much. I believe this is the end of this presentation. All right.